Alright guys, so we're going to keep moving forward and we're going to start talking about our next couple of taxonomic groups. And the next lesson is over protists and fungi. But we're actually going to split it up into a couple of days. We're going to cover protists today and then we will cover fungi on another day. And so protists are kind of a weird little group. In fact, of all of the kingdoms or all of the taxonomic groupings, I would take a guess that protists are probably going to be the least familiar to you. You know, we go through our daily lives and we come across bacteria, we see plants, we see animals, we see mushrooms and other fungi. But when it comes to protists, these are not necessarily things that we easily see in our day-to-day lives. It's not that they aren't there, it's just most of them are microscopic, and so they're just not something that we are as familiar with. And so your protists are going to be your first group that are eukaryotic. So we've already covered all of the prokaryotes. And so everything else that we're going to focus on is going to be a eukaryotic organism, whether it be unicellular or multicellular. And so this is going to take us back to a review of the endosymbiotic theory. And so if we break down these words, endo means inside, symbiotic refers to symbiosis or those close relationships. And remember that this endosymbiotic theory is the one that proposed how we got from our unicellular prokaryotic cells without nuclei and organelles to our more complex eukaryotic cells. And so it's believed that those mitochondria and chloroplasts that we see in our modern day eukaryotes were actually at one point their own independent prokaryotic cells. And so the whole idea is that prokaryotes engulfed other prokaryotes, and instead of digesting them, they lived together in a symbiotic relationship. Just like we have organisms that live inside of us today, we provide something for them, they provide something for us, and it's a win-win situation. So this is the idea of how we got these complex internal double-membraned organelles. And so the main evidence is really, there's a lot. When we look at mitochondria and chloroplast, they're about the same size as a prokaryote. They have a double membrane structure which supports that engulfing, kind of like you see here. They have these intermembrane structures. They have their own enzymes. They can replicate themselves inside of our cells. They have their own genetic material, which is circular, like what you will find in a prokaryote, and they have their own ribosomes. So they are essentially cells within larger cells. And so when we're looking at protists and fungi and plants and animals, they're all going to have these complex organelles that support this endosymbiotic theory. And so some of the things that would have had to happen in order to move from prokaryotes to eukaryotes is the cell wall would have to go away, because if a cell wall were in place, that would not allow for that engulfing, or what we call endocytosis. So it is important that the cell wall is lost, okay? Because otherwise a membrane could not surround and envelop another structure, okay? Now without a cell wall, we don't have structure. So first you have to lose the cell wall, which is going to remove structure. So you're going to have something that kind of takes its place. And so this is where we get the formation of the cytoskeleton. This is essentially a network of microtubules and microfilaments. These are just protein structures that are going to help support the cell. You'd have to have the formation of a nuclear envelope. It could just be some of those internal membranes, or the cell membrane kind of enfolds to create a vesicle. And then the DNA just happens to be attached to that, so that kind of forms that basic nucleus. You would have to have some vacuoles, or some type of digestive type of membrane sac that would allow for that engulfing. And then you would also have the formation of those mitochondria, which would lead to the ability to perform cellular respiration, take that oxygen and take that glucose and turn it into ATP. And so we definitely see a progression. This would not have happened slowly. This would have happened over a long period of time. But these are just some of the events that would have had to happen in order to create these eukaryotic cells that we see today. Now, when it comes to the chloroplast, there's actually different levels of endosymbiosis. You have primary, secondary, and tertiary. So earlier in our prokaryote lesson, we mentioned these cyanobacteria. These are in domain bacteria, and this is a group that is believed to be the ancestors to our chloroplasts. So primary endosymbiosis was when a prokaryote engulfed another prokaryote, or those cyanobacteria, and that would give rise to our modern day chloroplast. Now, secondary is when a eukaryote could engulf a green algae. Green algae is going to fit either in a plant group or in a protist group. And so literally it's a eukaryote engulfing another eukaryote. And then tertiary would be a dinoflagellate taking in another protist. So once again, we have a eukaryote taking in another eukaryote. So your primaries would be kind of how that origin of chloroplast inside of our cells came about. Now, when we talk about protists, once again, they're kind of a weird group. They're what we call a misfit group. At one point, they were a kingdom. In fact, freshman year, you probably learned kingdom Protista. But kingdom Protista is not an actual kingdom anymore. As we've mentioned, taxonomy is constantly changing. And one of the things that has changed over the years is kingdom Protista. So even though we kind of cover a six-kingdom system, it is not really a six-kingdom system. I don't even know how many kingdoms there are, because I don't know that there's agreement on how many kingdoms are in Protist and other groups. So for now, what you need to know is that it is a group formerly known as kingdom Protista. Now, what we're going to look at are the general characteristics. And honestly, the protists are the least defined of all of our kingdoms or all of our groupings. The only thing that they all have in common, once again, let me repeat that. The only thing that all protists have in common is that they are eukaryotes. So this is definitely something that sets them apart from the bacteria and the archaea, is that they are eukaryotes, which means they do have nuclei and they do have membrane-bound organelles. Now, the problem is they are eukaryotes that don't fit somewhere else, which is why we often called them the misfit kingdom. Because what we're going to see is that there are fungus-like protists, there are plant-like protists, and there are animal-like protists. But the only thing they all share is that they're eukaryotic. They vary in structure and function, and most of their commonalities do have the exceptions, except for the fact that they're eukaryotic. So here's some of the things that they may have in common. Once again, there could be exceptions to the rule. Most, okay, that's an important word, most are unicellular, which means they are one cell. Typically they are microscopic, and they're going to be found in areas where we can't easily see them because they are tiny and unicellular. But there are some protists that form colonies, like we saw with bacteria, and there are some protists that can be very large. Some of your algae groups can be very large. So they could be multicellular, but the majority of them are unicellular. Most of them use aerobic metabolism because they have those mitochondria that allow them to perform cellular respiration. There are a lot of different modes of nutrition. Now, this goes back to our last lesson. We saw terms like photoautotrophs and photoheterotrophs and autotrophs, heterotrophs, photochemo. So when it comes to your protists, because they're plant-like, they're animal-like, and they're fungus-like, they have different modes of nutrition. And this is really kind of how we group them within this grouping. And so what we have are animal-like protists, which are going to be heterotrophic. They're going to ingest things. Sometimes those are commonly called protozoa. You could have fungus-like protists that are absorptive. A lot of these are decomposers that break down material and then absorb the nutrients that they need. They don't really have a fancy name. And then you could also have plant-like protists, which we commonly call algae. And these are the ones that are going to be photosynthetic, and they're able to produce their own food using sunlight. When it comes to movement, most of your protists are able to move around. They are motile, okay? Some of them have flagella. Some of them have cilia. Remember, cilia is a eukaryotic thing. Some of them are able to move using something called pseudopods. Now, these are called false feet. Your amoebas, you've probably heard of an amoeba before, but your amoebas form these pseudopods, and what they do is they kind of push their cytoplasm up against that wall, and those pseudopods not only allow them to kind of creep along a surface, but they also allow them to surround and engulf particles by endocytosis. So this is one of the features of some protists. When it comes to their life cycles, it is varied. It really just depends on the type of protist. Because they are eukaryotes, they are able to perform mitosis. They are able to copy their DNA, divide their nuclei, and divide their cells to make more. Some of them are completely asexual, like we saw in bacteria, and what you'll see is pretty much copying DNA and dividing into two. But there are some asexual methods. Once again, it just depends on the protist. There could be types of fission where they divide into cells. Sorry, these are all examples of asexual. I think I'm getting ahead of myself here. So they could literally just copy and split, kind of like what we saw with bacteria. But because it includes a nucleus, what we see really is mitosis. It could be budding, where kind of a new one kind of grows off of the original and then pinches off and forms a new cell. Or they could form spores, like we'll see in fungi, that can develop into new organisms. Now, there are some sexual modes of reproduction as well, and once again, we're talking eukaryotes. So that could involve meiosis and syngamy, which is where the gametes come together. Remember, meiosis produces our egg and sperm. It really depends on the type of protist as to the timing. We saw back in meiosis that the way the life cycles work in plants versus animals versus fungi can be different, and so the same thing is going to apply here. Some of them have the ability to conjugate, which is not really reproduction. We saw conjugation with bacteria. It's where they exchange genetic material through a hollow tube. The same thing can happen with protists, except for in this case, the conjugation isn't necessarily an exchange of plasmids. Pretty much what they do is they kind of exchange nuclei, and that kind of shakes up the genome a little bit. Some of your protists are able to form cysts. A lot of these may be parasites, and so what they can do is they can outlast harsh conditions and kind of wait for better growing. And in the case of parasites, that can be a bad thing. When it comes to habitats, most are aquatic. Now keep in mind, most of these are unicellular, so they're not really going to be able to live in dry environments or the cells would dry out. So typically they are either in water or they are in areas that have moisture, so like damp soils or muds, things like that. A lot of times they do live at the bottom of a body of water, like those amoebas. They can kind of creep along the bottom of a pond or something like that. Many of them are components of plankton, and so plankton by definition are just tiny floaters. Now we actually have two types of plankton. We have phyto. Phyto refers to plant-like, and so your phytoplankton would be autotrophic. They are photosynthetic. You also have zooplankton, zoo or zoo referring to animals, and so these would be plankton that are heterotrophic, okay, because they're animal-like, so they would be ingestive. So plankton in general could be plant-like or animal-like. And plankton are essential to food webs, like in water bodies, and so a very, very important ecological role. Some of our protists are symbionts. They could be mutualistic, but they could also be parasitic. They could survive off the nutrients of a host. They could be definitely detrimental and have some negative consequences. It just depends on what type they are. And so what you're going to do as you go through your lab, okay, I want you to notice this down here. We're going to revisit these in lab. Is we can kind of take our protist and we can break them up into 5 major clades. Remember, a clade was an ancestor and all of its descendants. So we have 5 major groupings. We have alveolates, we have excavates, straminopiles, rhizaria, and amoebozoans. Okay. Now I know this is a lot to take in, and on your test you aren't going to see a lot of specific questions about these, but we are going to take a look at examples of these in our lab. Okay, like we're going to look at dinoflagellates. You're going to have an example of a ciliate, an apicomplexan. You're going to see euglena, diatoms, let's see, foraminiferans. I don't know that we're going to look at any slime molds or anything like that, but the main thing I want you to see is that your protist can be divided into many other groups. So what once was a single kingdom has now been broken up. And these are just those same group names that we just saw, okay? So in our lab activity, we're going to investigate these further and try to see some examples of these because they are so diverse. We don't really have time to go into the details of all of them individually. And this is where we're going to stop for today on this lesson. We're going to pick back up with fungi, but we are actually not done with protists just yet, okay? In fact, I'd like you to take a minute and I'd like you to get out the lab that you got the other day, okay? The one that looks like this. And so this lab kind of represents our three main groups that we're looking at in this unit. We've got prokaryotes, and hopefully you've already started, if not completed, your research on your bacteria that cause disease. Okay, once again, for these, you were just researching and just trying to identify what diseases these particular bacteria cause. And then notice we're going to have a protist section and a fungus section. And so I want to scroll down. So once you get your lab out, you have a section on protist, and you can see that each one has a circle, and then it has some questions. Okay, so I'm going to keep scrolling, keep scrolling, keep scrolling. Okay, and then you also have a section of fungi at the end. So what we're going to focus on today are the pictures for the protist. Okay, and you can see that they're broken down into alveolates. Okay, so you have some alveolates here. You have an amoebozoan. You have a paramecia. You have serradium, which is a dinoflagellate. You have plasmodium, which is going to be a disease causer. You have some excavates. You have rhizaria and straminopiles. So all those weird words that we kind of mentioned, we're going to look at some of those examples, okay? Now, for future reference, you are going to have a lab quiz. And this is probably going to be different than any lab quiz that you've ever had because you're going to see pictures, okay? And so what you are going to be expected to know from this lab are what do they look like, and you need to know the information about them, okay? So one of the things you could be asked was, here's a picture, name the protist, okay? Or you could get a question about that protist. So like I'm going to show you a picture of what an amoeba looks like under the microscope. And so on the quiz, you could see that picture and it could say, how do these organisms eat and move? So part of this lab is going to be the picture, which we have microscopes for, but what I found is that students often weren't seeing the same things. And so just to kind of keep it simpler and easier and to make sure it was uniform, I'm going to show you the pictures that you need to draw, okay? And we're going to do that here in a minute, which is why I needed you to get your lab out. So there's also going to be some kind of ecological significance about these protists. So like for instance, some amoebas can cause disease. And so here's a couple examples, and what you're supposed to do is kind of research and what disease do they cause. So when it comes to your lab quiz, for protists and fungi, you're going to be shown a picture and then asked a question about that picture. You could also see a matching section where it matches the name of the protist to the information about it. And so pretty much whatever's on this lab is going to be what you need to know for that lab quiz. And so what we're going to do now is we're going to take a look at these pictures. Okay, I'm not going to post the pictures on Google. The only place you're going to see them is in this lesson here. So you will have access, but I'm going to encourage you heavily to draw them so that you have the picture and the information side by side, okay? I would also make some notes. So when you take lab practicals in the future, a lot of times you will have pictures or microscope images that you have to look at. And honestly, for me personally, the best thing I could do is associate it with something that I know, okay? Or come up with something silly that helps you remember it. And I'm going to give you some examples as we go through the pictures. All right, so hopefully you have your lab ready, okay? And we're going to be starting right here. The pictures that we're looking at are going to be in order, okay? And we're going to go ahead and fill out our protist pictures, okay? So we're going to start with these. And so this right here is a picture of an amoeba, okay? I do want you to know that the colors for this activity, you might want to note the color, but please know that amoebas are not pink, okay? The colors are due to stains that they use to better visualize, okay? All of these are microscopic. All of these are microscopic images. A lot of these are way better than the microscope images that we would see. Some of them are similar though. If we were to put an amoeba under a microscope and look at it in lab, this is pretty much what you would see. All right, now I mentioned maybe make notes of things that it reminds you of, okay? So like for example, an amoeba to me kind of looks like a fried egg, okay? You can see that nucleus, which kind of looks like the yolk. Here I can see these pseudopods, okay, extending. And every amoeba that you look at is going to look a little different because of those pseudopods, okay? So they kind of take on different forms and shapes. So for now, you're just drawing the picture in that circle, and then later you're going to have time to work on your lab at another time. You're going to do the research portion. So like what diseases can these cause? How do they move and eat? All right, so go ahead and draw your amoeba. The next picture, this is our paramecia or paramecium. Okay, these are an example of a ciliate, and you can kind of see those little cilia, those hair-like structures going all the way around. All right, paramecia kind of beat those cilia back and forth, and it allows them to kind of swim in a water body. Once again, you can see the dark nuclei. All right, and you're going to have some questions that you need to answer about paramecia. All right, moving on along. Okay, our next picture, this is going to be our dinoflagellates. Now, an example of something silly, this one kind of makes me think of pterodactyls, which are a type of dinosaur. And so dinoflagellates are actually responsible for kind of a negative ecological event. That's one of the things you're going to be researching. So for now, you're just going to draw them. You might want to make note of their color. Once again, depending on what microscope slide you're looking at, they could appear in different colors, okay? So kind of do your best to draw what they look like, and then maybe jot down some things that it reminds you of or something that might help you connect those two together. All right, I also want to remind you that on the quiz, when you see the picture, the question could simply be, name the protist, or the picture could ask a question about that, like, how do these protists move, or what disease do these protists cause, okay? So just kind of keep that in mind as you're doing your research. All right, we're going to move along to our next two protists, okay? And notice that these two slides look kind of similar. Now this is a microscopic image of Plasmodium. This is a microscopic image of Trypanosoma, okay? Now what you're looking at are red blood cells, okay? Both of these are parasites that can get into the blood and cause disease, okay? So all of these little kind of pink or reddish looking tubes, those are red blood cells, all right? Like this big purple thing right here and that purple thing there, those are white blood cells. So what we're looking at is a blood smear. Now, in order to identify the protist, okay, in this picture, here you see an arrow, okay, and it's pointing to an abnormal looking cell. Here's another one that has an arrow, okay, it's pointing to another abnormal looking cell. Here's another arrow here. What we're looking at are cells that have been infected by Plasmodium, okay? So we're not necessarily seeing the protist itself, but we're seeing cells that are infected by the Plasmodium protist, okay? And this actually causes a very significant disease that is huge, not necessarily for us where we live, but in tropical areas, and it kills a lot of people each year, okay? So this is definitely ecologically significant. So once again, what we're seeing is a blood smear, and these are cells infected with Plasmodium. As we move down the center picture, this is a picture of a blood smear as well. And this is showing us the protist trypanosoma. And so trypanosoma are these little wormy looking organisms here. Okay, so there's a trypanosoma, there's a trypanosoma, there's a trypanosoma. All right, so both of these we're looking at blood, but we're looking at the protists that are infecting, okay, that blood. All right, the last picture here, these are pictures of euglena. Okay, euglena have a unique way of getting nutrition. They're pretty diverse, that they have multiple ways that they can get food. An example of what these kind of look like to me are, they look like little leaves. Okay, so euglena and leaves kind of go together, or anything else that it might make you think of. So just be sure you draw a picture of euglena there. All right, once again, they're all in order. So our next picture, this is a picture of diatoms, okay? And diatoms always remind me of like a kaleidoscope or like confetti. You can see that there's a lot going on this slide right here. And if we were to look at a slide on a microscope there in the room, this is very similar to what you would see. So you can see different shapes, like here's one that's kind of oblong. You can see different colors because they're stained with different colors. These are definitely some plankton components here. These are some of your tiny floaters. But they have a unique cell wall, and that's one of the things that you're supposed to be researching, is the components of their cell wall that kind of gives them this glass-like looking structure. All right, moving on down, we have one more protist that you need to draw today, and that is going to be your foraminiferans. Okay, and these kind of look like little shells. These are microscopic, okay, so these are blown up a whole bunch. And something that you might not connect unless I bring it up, but when you watch the video the day the Mesozoic died, it talked about forams or foraminifera, and it was talking about the fossils from these little tiny organisms before and after the extinction. And so this is an example of what your foraminifera look like under the microscope. All right, we're actually going to stop there because the rest of them are going to be fungi pictures. And so today was all about protists. So if you didn't get a chance to get all your pictures, be sure you get those in your lab. You do need to draw them. And once again, I would make some notes about what they remind you of. And then as we move forward, you're going to have a day to work on your research for both your protist and your fungi. And that's all I have for you guys today. I hope you're doing well. Let me know if you have any questions or concerns, and I will talk to you soon.